Welcome to Equinox, where Rob and I are striking the balance between the light and the dark. We are cranking the show up to 11. My name is Joseph Darnell, and I'm joined by my good friend, Dr. Robert Carter. Hello, Rob.
Hello, Joe.
It has been a wonderful weekend, and now on the start of a new week. It is so cold in Georgia. It is weird. I don't know why, but this time of year, I'm not drinking hot tea, but I want to get like all bundled up in like 3 quilts.
This is one of the coolest Aprils I remember, and it's just now. I mean, it was, in the 70s yesterday. It's plummeting down to under 50 probably. So what is happening?
It was in the 40s last night.
Yeah.
It's just not right. And like we went out to this beautiful gardens called Gibbs Gardens earlier today. Beautiful place, very expansive. All in all, it was about a 3-mile walk from the far end of the gardens back to the car. And beautiful scenery, but it was hard. We're there for the flowers. We're there for the manicured trees and beautiful creeks and ponds. But it was chilly. And we thought that our body temperature would warm up and we'd be okay, but it just never really got any better. We were a little shivering when we got back into the car.
It feels like October.
It looks like spring and it feels like October.
October is my favorite, but I'm just not ready. And it was just kind of weird. I'm sure this has happened every year of my life. I just don't remember the weather being like this at this time in Georgia.
I don't, and I have a pretty good track record of remembering because my birthday is in the middle of April, and if anyone should know, I should know.
Yeah.
And it's never like this.
And yet one of my cousins was showing the 6 or 8 inches of snow on her porch railing just last week, upstate New York.
That's awesome. We didn't get any snow this year that I can recall.
No.
And I miss snow.
It was sad.
We got all the cold weather, but no snow. What have you been up to?
Well, I spent the weekend doing all sorts of things. I finished up one beehive, and that was exciting. So I'm sitting on my front porch right now. And then in some extra little spare moments of sitting down, I did a lot of genealogy.
Genealogy?
Yeah, I was digging into some of the records in the old countries, and it's really kind of cool because with Google Translate and with open access stuff that's just on websites already, I can read in Dutch now, and I can read in Norwegian.
Okay. So what are you doing with these languages?
Well, I'm tracking down some of the old family members.
Oh, I got you.
And the Norwegian records are cool because they went, and the country grabbed all the old church records and digitized them. And since Norway hasn't been burned down with a giant war in the last 1,000 years, they've got some really good records. And I've got some families, or I've actually been able to find the little white church where my great-grandfather was born, baptized, where his parents were married, where his father was married. And so all these generations of records in this one little teeny place, in this little valley 30 miles from the southern coast of Norway in the middle of absolute nowhere.
Wow.
And I can read the records.
Yeah.
Because you learn 15 or 20 words, and they almost sound English.
Oh, okay.
Far as father and more as mother.
Oh.
And the coolest one I found, I'm looking at this word, it sounds like true love. True love. No, true love. It's the witness at a wedding.
Oh, interesting.
And so, these are people that these people knew. And sometimes it's my great-great-great-grandfather. Oh, he was at his kid's wedding. Oh, man.
Nice.
So, and in the Dutch record's even better.
I'm going to have to use that, by the way. That is a great term. The true love.
The true love. Wedding. Yeah, I like that. But the Dutch records, the most women in Dutch society kept their name. They didn't take on their husband's name.
Does that present a challenge?
No, it's great. Because it allows you to track the female side like you can't do in most other cultures.
Nice.
Most of my lineages, when there's a dead end, it's because there's a lady. I know her first name, and her last name is the name of her husband. You can't track that. You can't go beyond that. You're kind of stuck.
What were the cultural reasons for why they didn't change their names?
I don't know.
I don't know either.
I don't know. But they also, just like in Norway, in the Netherlands, especially in Friesland, up in the northern part of the Netherlands where I'm looking, they tended to name their child after the father.
Okay.
So like Jensen, the son of Jens.
Yeah.
Or Jens, I guess. There's no J.
Jens.
Anyway. And so you're able to look at genealogies and family records and just go up generations, and they have such wonderful records all the way back to early 1800s. Some of them I got down to 1600s in one branch in the Netherlands.
Any particular revelations?
Oh, yeah. My great-grandmother, Weibrigge, Wibrigge, something like that. I've tried to pronounce it, and I've had someone who is from there try to teach me how to pronounce it. I keep flailing. I can't do it. Her father and mother, I found this record with this. So her father and mother would be my great-great-grandparents. My great-great-great-grandparents are in a record, and all 4 of their names are there on both sides of the family, but the children are different. Oh, my great-great-grandfather's brother married his sister-in-law.
Oh.
Or 2 brothers married 2 sisters. And that messes up all the genetics.
Oh.
So if I'm on 23andMe.com, I've got a lot of relatives from Holland.
Explain why that messes up the genetics?
Because all of us have a double dose.
Yeah.
All these double first cousins, when you look at the genetics, they say, oh, you're this many generations apart. Actually, no, we're more than that. But we have more DNA in common than we should.
Oh.
Because the one brother came to America and the other brother stayed behind. And the one sister came to America and the other sister stayed behind. But their children are like brother and sister, even though they're cousins.
Yeah, so the same issue would arise today if the same thing happened today.
Oh, yeah.
2 sisters marrying 2 brothers.
And it happens.
Wow.
But I couldn't believe I found a record of this entanglement in my family tree.
That has got to be pretty rare. We were thinking about, my wife and I were talking about some friends that ran into the same arrangement recently. Interesting stuff.
And I know a girl in college, she was always very self-conscious about it, but her mom had died and her dad had married her aunt. So he married his wife's sister.
Oh, okay.
Which, is common enough. It's not like that never happened, but she was always, I am my own cousin. She was always very self-conscious about this.
Was she like a teenager or a preteen when it happened? I could see,
sure.
I could
I knew
understand
her when she was
in the
18,
right circumstance
19.
that
I
would
don't know how
make
old.
you pretty self-conscious.
Yeah.
Oh, come on. Yeah. That's not even a problem, to be truthful. So we wanted to talk about your background this episode. A little different. Everybody's wondering, who is this Dr. Robert Carter? I mean, we've already listened to you for 10 episodes, and we don't even know who this guy is. We started our journey with Isaac Newton. We learned about him. We don't care about him. We want to hear about Dr. Robert Carter. We want Charles Darwin. Forget it. We want to know who this guy is.
Only if the conversation is more science than it is biography.
Oh, okay.
Right.
But that being said, we talked for the last 6 minutes about your biography and your family tree. No, okay.
Oh, okay, fair enough.
In truth, though, you are a scientist, and we talk a lot about history, and we do talk a lot about your interest in sciences in general that aren't always your field. So I wanted you to take the mic and describe yourself, explain yourself, and how did you become a marine biologist?
The reason that we have all these subjects that don't seem to be related is because you and I.
Oh, they are totally related.
Well, in a lot of different ways, but there's not like a
not obvious.
It's not a
academic
one
ways.
subject
Yeah.
podcast here. It's because you and I are both interested in so many different things.
We work in an organization that dabbles in a lot of science topics.
I have been a science geek nerd since I can remember. Always been just really, really interested in how things work, why things work, really small things, really big things, really fast things, just because it's cool.
And they are.
They are.
That's why we have a podcast. Science is cool.
I ended up at Georgia Tech as a biology major, very strangely, not an engineering major. I'm not even sure how exactly happened. It's just one day I just got this thing in the mail and said, hey, Dad, I want to go to Georgia Tech.
Really? You just
Yeah.
got the itch to do it when we get that desire we cannot explain to do something.
But I lived 1,000 miles away.
Oh, really?
Yeah,
So then why Georgia Tech?
I don't know. It was
Man.
a total whim.
The marketing in that letter must have been pretty good.
I was looking at all sorts of local schools and, Rochester Institute of Technology and, up New England, New York, Midwest, farthest, Purdue, maybe the farthest away that I applied to. And I got into a lot of colleges, but I don't know, something about Georgia I think I'm going to go there.
I think it sounds like an adventure. As a young man,
Yeah.
you were attracted to, seeing new worlds and strange civilizations, going where no Robert Carter had gone before.
What's weird, though, is during this transition, I became a landlubber.
Okay.
I grew up on the coast. I grew up with a boat basin in my backyard because my parents owned a marina.
Nice.
I grew up on the water.
Does that explain the marine biologist?
Later on,
Okay.
I had taken a joint enrollment course with a local college my senior year, which is a marine biology program. And I really liked it, but it was just science because I was also taking physics and I loved physics, as you can tell from the podcast. And I don't know, I just decided I was going to take biology because my best grades in high school were in my biology class and marine biology. I just aced them. English, not so much. No, not even close. Anyway.
That's not a science anyway.
No, we don't talk about English. I had to learn how to read and write later on in life, shall we say. One of my guidance counselor was very mad at me when my SAT scores came back because my English and math scores are only 10 points apart.
Oh, no.
And he says, Rob, we have a problem. I said, what's that? He goes, you make straight A's in math and you make straight C's in English. Why are your points so close? I said, I don't know. I think it's just lazy.
Yeah,
But
speaking for myself, I knew my way around Photoshop when I was 12, but I don't think I read any books that decade. I'm like, I'd read a chapter now and then because I had to, but I don't think I finished any books over 100 pages long. A couple of years later, I wrote my own fiction, though.
oh, yes, cool.
I got into that. I was a homeschooler.
Ah, yes, the old prodigal homeschooler. Coming back to love of learning. I was nearly 20 years old where the love of learning kicked in.
It was gradual for me, too, and honestly, a lot kicked into high gear for me around the same time.
If it had happened earlier, I would have had much better grades, and I probably would have been an engineer. I didn't get into Georgia Tech's engineering program. My grades in high school weren't high enough. And so they gave me, I was a provisional student, and it was while I was there, I got challenged by one person in particular, who I won't mention, but one person in particular.
Or teacher?
A student who was also a co-worker, because I worked at the state EPD alternate semesters to pay my way through college. And I remember one day, he's a nerd and I was a nerd, and he was challenging me on stuff I didn't know. And he walks up to me, slaps a piece of paper on the desk. He goes, Rob, all 50 states and capitals, go.
But why would you need to know?
Just a random thing. Okay. I had never studied geography. I knew nothing. In fact, there's 100 answers, right? 50 states, 50 capitals. I got a 50%.
College nerds are brutal.
And I was humiliated. I missed like Nebraska, Iowa, this whole strip of states. My United States it wasn't wide enough.
Yeah.
Because I'd never been in those places. I have been there now.
Sure.
And so that semester, he and I memorized all the states, all the capitals, the Canadian provinces and territories, the Central American states and capitals, South America, Europe, Africa, Asia, and we ran out of semester when we got into the Pacific Islands.
Okay, can I inject here?
Yeah.
I was homeschooled, so I knew my way around geography. No, I'm kidding. But what we did have was I went to this friend's house while my parents went to a parents' homeschooling conference, 2 years. And at this friend's house, while we were there, while my parents had the conference, we were at the friend's house. She had this smart electronic globe thing that a lot of homeschoolers probably had, but it was like a geography quiz trivia machine. And so It was gamified. And you turn it on, bloop, 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 bloop. Do you want to be quizzed on America, the Americas, on the whole world, on certain continents? And I played that game and learned all of geography because of it.
Cool.
If you had had the same thing, I'm sure you would have known it like the back of your hand.
I would have eaten that up.
It was a great
After
toy.
I was 20. If I was 15 or 12, I would never have touched it.
Really? See, I was like 9 years old and I loved it. It was a pretty boring week, at a girl's house.
What? You spent a boring week at a girl's house when you're 9?
Yes.
What do you mean?
Well, my parents were at the conference and we were there at my friend's house. And
okay, I see. Okay,
it's not like she had any pop guns. It was a girl and she just had a playground in the backyard and this one toy globe.
I'm sorry that you're so scarred.
Everything else in the bedroom was pink.
Oh, that's awful. Sorry, girls, I'm sure you love pink. Some guys do.
No, I like pink now.
So here I am. I'm being challenged intellectually for really the first time, and I'm loving it. I started really getting, especially the influence of this one person still. I started getting hugely into military history and geography, and I'm a science major, so I'm learning all this stuff about biology, and I'm loving it. And all the other science classes that I had to take. The 1C that I made at Georgia Tech was in English, so I actually missed highest honor because of 1C.
Oh, wow.
Of course, if I had made an A where I made a B, that I also would have gotten it. But that one English class, that just sticks like a bone in my throat. Just grrr.
Oh, that's so not fair.
But now I am an author, and now I know how to parse sentences and put periods in proper places. But it's been just a lot of work to get there.
I've read a lot of your articles. They're pretty good. I like them. I honestly enjoy reading them.
Yeah, I enjoy listening to you read them.
Right. There'll be a link in the show notes.
Excellent. So I'm done with Georgia Tech, and I go to become a high school teacher. And I spent 4 years teaching high school. My first job out of college was teaching summer school chemistry. And I thought after 3 years of Georgia Tech chemistry, I made straight A's in organic chemistry. I loved it. Everyone else was like, this is terrible. But I loved organic. I made B's and C's, A's and B's in the rest of the chemistries. I was like, I can handle it. No problem. And it almost killed me. Oh. Summer school chemistry. I was cramming to 2 or 3 o'clock in the morning every night so I could walk in the classroom at 8 o'clock in the morning and pretend I knew what I was talking about.
Oh,
But I learned it.
yeah.
It's a different thing taking a test versus having to explain what it means to somebody. And I fell in love with teaching. And I taught AP biology, I taught physics and chemistry and electronics for 4 years. And it was a dream job.
I think that is the best way to teach, is teach your students to be able to explain it to somebody else.
What I figured out was when I was able to explain it, that's when I knew it.
Yeah.
And if I couldn't explain it, I did not know what was going on.
I think that helps students to appreciate that they actually accomplished something with any lesson of any subject. You said it was a hard summer, but that just makes so much sense. I mean, the sense of the accomplishment has to be out the wazoo.
Yeah, actually, we should do a show on chemistry. I love chemistry.
Yeah,
Maybe
definitely.
We'll kill our audience, but who knows?
The queue is getting pretty big. I'm going to put it down around topic 26.
Yeah.
Okay, chemistry.
And we're going to have other people, hey, you need to talk about this. So that'll inflate the queue a little bit too. Mm-hmm. That's fine. It's always good to have more materials that we can do.
Yeah, we'll have to figure out a way to do a poll with the audience and let them vote on what to move up in the queue.
Yeah. But now here's where the marine biology comes in. It's a long story to get to this point.
Oh, yeah.
While I was there.
You met a mermaid.
No mermaid.
Oh, okay.
The Parents Association bought a very large aquarium for my classroom. And I put salt water in it and live rock and fish and corals. And that was my first saltwater aquarium. Later on, I built an aquaculture facility at the University of Miami for Caribbean corals. So I had tens of thousands of gallons of salt water and thousands of coral colonies under my care eventually. But
excellent.
At the time, it was one aquarium, and I'm asking questions. How does this work? What's that thing right there? You know, how all these questions. And I realized that a lot of the questions I was asking, no one knew the answer to. I said, oh.
So when you say nobody knew the answers, you mean nobody there did, or nobody really academically anywhere?
Yeah. So there's questions that you could ask by staring at a saltwater aquarium that don't have an answer yet.
What year was this? What time-ish?
That was 90. I left in 96.
Wow.
To go to marine biology school at the University of Miami. Well, what I figured out was I'm interested in too many things. My brain doesn't settle down on one subject easily.
That's one of the things that I was wondering was, how did you pick marine biology? Because science has so many interesting subjects.
I figured it would be interesting enough that I could study it for 3 or 4 or 5 years and I would survive and not be bored stiff. It took me 8.5 because I wasn't a scientist. I was a nerd. There's a big difference.
Oh.
I didn't know how to do science. I knew how to take tests. I didn't know how to design an experiment. I didn't know how to run a program. I could follow a recipe. At first it was just classes. I was taking classes. I was doing great in my classes, but I was supposed to be starting my research program and I had no idea what to do or how to do it. And then my professor left in the middle.
Oh, okay.
But that was the best thing that had happened.
Oh, good.
Because I was in the coral reef ecology program, but I was not a very good ecologist. And I was getting really frustrated with it because just theoretically, I wasn't doing well with thinking like ecologists think, but also numerically. They were coming up with numbers. They're trying to put numbers on God's creation. But creation is so incredibly complicated that most things defy easy numbers.
What do you mean by numbers?
If I capture so many tons of fish and off the coast of Peru every year, what's the carbon budget and what's the population going to be the next year? That depends on the temperature. It depends on the direction of the currents. It depends on a million things we don't know. And so they're doing all this modeling, all these complex ecosystems, and none of the models worked.
Oh.
And granted, they're getting better and smarter, but at the time I was like, this is really ridiculous. And so
ridiculous that y'all didn't know, or ridiculous that they were even asking, or?
I was getting frustrated in ecology because it wasn't a hard science. It was too loosey-goosey for me, and there was too many numbers I didn't know. Some people are really interested. Oh, we can really get into this. And I was just like, I don't want to go there. But that was my academic program, and what was I going to do? Well, after I was left with no lab, no support, which wasn't bad. I still had an advisor, and I still had office space and a laboratory, but I need to reinvent myself. So I spent 6 months writing grant applications. And the 2 applications on coral reef ecology didn't go through, but the one on genetics did. And it was actually funded through the university.
Oh.
I was working with corals. I was doing hundreds of scuba dives. It was a tough life. I mean, twist my arm, got to go down to Florida Keys again. And it was like 3 times a week all summer long for 4 or 5 years. And it was just a dream job. It was wonderful, especially during coral reproduction season. It was an awful lot of work and it was very little sleep, but we would go out before sunset and anchor and watch the sunset. And then after the sun went down, we'd scuba dive because 3 hours after sunset, 4, 5, and 6 days after the full moon in August, the major species of coral on the reef would spawn. They release eggs into the water. Billions and billions of them. And then 7, 8, 9 days, another massive species would spawn. And so we were out there collecting eggs and sperm and doing, breaking colonies off and preserving them and looking at them under a microscope to see what the developmental stages were. And it was really cool, but it was late night work. And then if they spawned, of course, you're trying to capture the fertilized eggs. And every year we tried to raise them and we always failed. Because a coral that could, as big as your house starts out as something that looks like a speck.
That was always something I thought about nature documentaries, watching them growing up, was I couldn't really appreciate the scale of the coral reefs. And then they would like tighten in on a few fish that live in and around the coral in a nook or cranny. And then they would show this wider shot where you could see the depth of the ocean, the coral with some depth, but you couldn't really tell, is that in yards or feet? You just couldn't tell.
Yeah.
Or if it was 3 stories deep or what.
Yeah, you get lost with some of the size, even when you're up in the middle of it. But you get used to it, and I would regularly see coral colonies bigger than me, regularly, all the time. In fact, one time the Park Service had put a new buoy, a warning buoy, in what's called Hawk Channel, which is from Key Largo in the Florida Keys offshore about 5 miles. That's where the reef is, about 5 miles offshore. And between the reef and shore is called Hawk Channel. And they put a new buoy in the channel and said, oh, there must be a patch reef there, a shallow spot that someone had run aground on. And so we suited up and jumped over with scuba, and it was just this huge coral colonies. I mean, all the way up to the water surface almost. They couldn't grow taller because low tide, they would have been exposed. And we're swimming around this thing, and it was literally as big as a house when I realized this was one coral.
Wow.
This so-called reef actually was one animal that had been living in this one spot for probably 1,000 years or more. And it was huge. I mean, it was massive. It was unbelievable that one animal could get that big.
Wow. I thought I knew things about coral, but a coral living for 1,000 years?
They can live. They can theoretically live forever. But that's another mystery that they have, because
it's not like they have rings on the inside of a tree for coral.
No, some yes.
Really?
Because they grow slower at sometimes and faster than other times. And if you slice their skeleton and polish it flat, and you look at it under a microscope or you take an x-ray of it, you can see density bands. The little skeletal components are thicker in some places than others, because some places are growing fast and some places are growing slow. But it's not clear if that's summer or winter.
Yeah, it doesn't sound super precise.
It's not super precise, but you can do it pretty well for a lot of species. And it's really a cool science when you do that.
Interesting.
But those animals, even if it's as big as a house, it's really thin. It's only, I don't know, half a centimeter in thickness. So it's a single animal with 1 million mouths. They say it's a colony. It's not a colony.
Right.
It's one animal. It's not like a colony of penguins.
I've heard that a lot.
It's one animal with lots and lots of mouths, because if you're thin and you want to get big, you have to spread out like a sheet. But if you spread out like a sheet, well, the end of you is far away from your mouth. So you make another mouth.
It's just that simple.
It's that simple. And just beautiful animals. I spent a lot of time staring at them under microscopes, specifically fluorescent microscopes, where you shine ultraviolet light and you're looking with a filter so you don't burn your eyes out through a microscope, and you're looking at all the beautiful fluorescent colors. Or you shine blue light, and then you put a yellow filter on the microscope so the blue is filtered out, and now you're looking at the green fluorescence from blue light. Or you shine green light and you're looking at the red fluorescence. And that was actually my PhD dissertation, was on the fluorescent proteins in corals.
I wondered about that because of your other fluorescence project where you made glow-in-the-dark fish.
Oh, yes, exactly.
Like, I wondered how you got to that.
Well, I was working on the coloration of these animals, because ecology talks about weird things like coloration patterns and things. I was trying to put numbers on it, and I consistently failed. So my dissertation had 3 chapters of nonsense, where I did lots and lots of, it wasn't nonsense, I did lots and lots of experiments and I proved nothing. I could not get them to change color. I couldn't get the color to be brighter or dimmer. In fact, one time I walked into our invertebrate museum, which was at the end of a hallway where my office was. And I went in there and talked to a lady. I said, do we have any deep water corals? She goes, oh, yeah. They had some that they had an expedition back in the 1930s. They had dredged up some corals from 2 miles in depth.
So if we're talking about 1930s corals, I'm assuming they're long since dead.
Yeah, they're in alcohol. They're sitting in literally a pickle jar with a metal lid since the 1930s.
Well,
and I might have been the first person who would open up that jar. Oh. And I took a syringe and I pulled some of the stuff out and I looked at it and the alcohol was green. These things had green fluorescent protein in them. And I put it in a spectrophlorometer and had exact same signature as my green fluorescent protein from shallow water corals. So I said, oh, this has nothing to do with sunlight. So the biggest hypothesis, these things are green. They're absorbing these harsh colors of light, ultraviolet light, and are fluorescing it at longer, safer wavelengths. It's photoprotective, everybody said. But then I said, then how come this animal that lived 2 miles in depth has got so much in it that it actually colored the liquid that has been sitting in for 70 years? It's not photoprotective. I never found out what the function was. I listed about 17 different hypotheses of what this stuff might be for and 17 contradictions. And so I'm a very frustrated scientist, but I'm working on this stuff. I'm doing chemical extractions and I showed that it was a protein. That's bizarre because we're made of proteins and we don't glow in the dark. Right? We're not fluorescent. You had to go to a rave or disco or something. You had to wear fluorescent makeup or clothing to when they turn a black light on for you to look funny colors.
Laser tag.
Oh yeah, laser tag, yeah. But we don't do that. But all the cnidarians, the corals and jellyfish and the sea pens and all those weird stingy things in the ocean, they all glow. Well, some of them are like a firefly. Some of them produce light.
Oh, wow. That's a whole new level. I didn't realize it could go that far.
Oh, man. I remember as a kid on Long Island at summer camp. It's funny because we were only a mile away from the local nuclear power plant. Interesting. But we're fishing at night. And you cast the line and you can see your fishing line going down in the water glowing green.
Oh, that's fun.
I think this is from diatoms, bioluminescent diatoms. One time I had a massive poison ivy on my leg because I'm stupid and I didn't think of stay away from the poison ivy, Carter. You're going to have to go to the hospital again. But I had this massive, my whole entire thigh was one giant poison ivy thing. My friend he also got poison ivy. And we were camp counselors and the camp director said, salt water is good for it, boys. After you put your boys to sleep, because all the counselors, there was always one counselor on duty and everyone else could leave. We went for a swim. And so we swam out to the platform and he's in front of me. His name was Rob too. Okay, Rob, you're glowing. It's like, what? And he stops and goes, no, you're glowing. And we looked at each other. We could see our legs and our feet because the water was full of, I think, diatoms. And so what we did is we swam out to the swimming platform. We jumped off and we watched each other go poof in the water. And you could literally see two hands and then bubbles. Oh, it's the coolest thing. And so now I know there's some bays in Puerto Rico still that glow. They're bioluminescent bays. There's so many bioluminescent organisms there. The water's literally lit from inside.
So when you say diatoms, did you tell us what diatoms is? But when you brought it up, in that context, you had just talked about your fishing line in lure. So I thought diatoms was like a brand of...
A diatom is a little alga. And it has a little crusty shell. And they're tiny and they're beautiful under a microscope too. And some of them are bioluminescent. Some jellyfish are bioluminescent. They produce light, but the light they produce is not green, it's blue. But they have this protein that absorbs blue light and fluoresces it at green light. It's called the green fluorescent protein. And that's what my PhD dissertation was on. So there was a guy working on fish and he had green fish. And he's in a building across from my building. And someone said, hey, you need to go talk to that guy. So why? He goes, because he's got green fish, like fluorescent green fish. Well, I'm working on fluorescent green. I learned that protein he was using came out of a jellyfish. I'm working on green from corals. Corals and jellyfish are similar. And so I went over there and we started talking. And he said, this is the same stuff I'm working on. But corals are also red and yellow and blue. Oh, so he got very interested. And so we worked up a protocol where even though we knew that other people were looking for these things, and it would take them about 2 years, we said 24 hours. And we approached the technology department at UM, the patent office, and we said, we want a grant. We can do this work and we can blow the doors off everybody else. And that's where my grant came from, the long shot. That's why I became a geneticist.
Wow.
And it was the best thing ever because I'm a good geneticist. I was a lousy ecologist. I'm a good geneticist. I love genetics.
Nice.
And the genetics I'm doing now, I like even more because computer programming genetics. But back then it was extracting DNA, purifying DNA. What we did was we reasoned that if there's an animal that's glowing under ultraviolet or blue light, it must be producing a lot of green fluorescent protein. If it's producing a lot of green fluorescent protein, we don't know where that gene is in the genome of the coral, but he must be producing lots of messenger RNA, because the messenger RNA is what is translated into protein. So we said, oh, let's extract all the RNA from the coral and throw the DNA away. And then we reverse transcribe the RNA into DNA, put that DNA into bacteria at random. All the RNAs float around in that coral are now in E. coli. And we put it on a plate and put it in an incubator. And we said, if we're right, 1 out of 10,000 colonies on that plate will be green. We were wrong. It was 1 out of 1,000.
Oh.
So we had a plate, and you do a dilution where you have a lot of bacteria in this one, and you divide it by 10, divided by 10, divided by 10. And by the time you're at the end, there's like no bacteria. And somewhere in the middle, you have a plate with individual dots where they're not right next on top of each other. And if you have an individual dot, you can actually take a little needle and pick it up and then spread it on another plate. And you have a plate that's a complete clone of one bacteria.
So would there be a practical application for making bacteria's bioluminescent?
The green fluorescent protein family has turned into one of the most important tools in biotechnology because it's a protein you can see.
So it is very practical.
It's incredibly practical, incredibly important. When I was leaving, they were taking the fish that we had made and they're trying to develop a cancer model where if the fish got cancer, the tumor would be green. And when the fish was cured, the green would go away. So it was visual genetics, which is unlike anything else. It was very difficult to do, and I don't know if they actually succeeded because that was 2003. But they had attached the protein to 2 different genes. One was called a heat shock protein, where under stress, this protein turns on. If you put the fish in water and warm it up or stress the fish somehow, it would turn green. The problem is it was always a little bit green. But they also got it next to a gene, it was called a metallothionein. It's a protein that grabs toxic metals. Zinc, cadmium, things like that. And what they did was if you had a fish in a fish tank that wasn't green and you added some zinc into it, the fish would turn green.
Oh, that is neat.
Cool, but it took a long time for it to be ungreen. Because once you have protein, it's there and it takes a long time for it to go away. So it wasn't an instant on-off switch. But they're working on stuff like that and it was really cool. But I also said, well, okay, now we have the green. Let's go for the red. Let's do the same thing. So I had been in an aquarium store.
On that note,
yeah,
something I know about lights of different colors is that green light is the brightest of all the colored lights.
Yes.
So I know this is really nerdy.
Do you know why it's the brightest? Of course you do.
I'm gonna sound really nerdy for noting about this.
Nerd out, buddy.
All right, Star Wars. We all know you got your blue lightsabers, red lightsabers, green lightsabers, and then one day out of the blue, there's a purple lightsaber that just one Jedi Knight has, and nobody else does. But then in some of the movies, they look white, and then other blue lightsabers look more blue than others. Okay, you ever heard of these things called what are they called? Dragon Con, Comic Con, and
yeah, there's a massive one in Atlanta every year.
Okay, when you go, you got to be in costume, and you've got to have a lightsaber.
Oh, you got to. Okay.
So this means that there is a business for finely crafted lightsabers.
Oh man, I missed out on my calling.
And they can be in, what do they call them? Like aircraft grade metals.
Okay.
Used on jet airplanes.
Okay.
Indestructible housing for attachable, detachable blades.
Okay.
And come in any color you want. And I remember discussing this stuff with Park Sabers. I think the guy's name was Jack or Jason Park.
Okay.
So parksabers.com, like over 10 years ago. Okay, so tell me about your lights and your blades. And he explained that the green was the brightest. And then it went down from there to the darker colors like purple.
But it wasn't brighter because it has more power.
Right.
It was brighter because of the biology.
But I didn't understand that. I don't even know if Park told me that. So, but I just knew like there was green, then, yellow, then orange, blue, and then reds and purples. It just got darker and darker as it went.
It's because the human eye is most sensitive to green light. And look at our world. Our world is green.
I didn't even really think about that.
God gave us a green sensitive eyeball because there's so much green in the world.
Interesting. So does that mean that of the other colors, we could look at a list of what colors our eyes are most sensitive to, to least sensitive to?
Oh yeah, absolutely.
So do you know what a few of the other top colors are and what order they are?
No, it's green is the peak, and it falls away towards the blue, and it falls away towards the red until it disappears into infrared or ultraviolet. You can't see those colors. Now, birds and bees might be able to see some of the ultraviolet colors that we can't.
Right.
But that's just where our eyes are tuned for.
Wow.
Even though we have 3 different color receptors, the green one is the strongest color receptor. And interestingly, we have these green fluorescent proteins, and green penetrates furthest in water. Red is absorbed very quickly. One time I was diving, and I was about 60 or 70 feet, and I put my hand on a rock, and all of a sudden, wham! Ouch! And I hollered underwater. I'm shaking my hand. I don't think I was swearing, but I was hollering really loud. And I looked at my finger, and there's black goo coming out of my finger. And I'm like, I've been poisoned. I've been poisoned. Oh, and I'm hollering, and it hurt. And wait, hold up, Carter. Don't be dumb. You're 60 feet. There's no red light at 60 feet. You're looking at your blood, and your blood is black.
Oh, that is creepy.
Oh, yeah. And so I looked over the side of the rock. I'm like, what got me? I'm looking, and I see something about the size of a quarter, maybe a little bigger. It's a mantis shrimp. And what I'm looking at is the top of his burrow. He might have been 4 or 5 inches long, but he digs into the sand, and he sits there with his head right on the edge, and he flattens his claws out and over the hole, and so it looks like it's flat. And so I picked up a stick or something that I found, and I kind of poked at him, and I watched how fast he moved.
They have the fastest known, punch of any creature in the world.
The pistol shrimp moves his hammer so fast, it actually breaks the speed of sound and makes a flash of light.
The mantis shrimp can do this too.
Okay, but the pistol shrimp you can hear.
It would probably sound like a gun.
Yeah, pop, pop, pop. But the mantis shrimp just hurts. Oh, man, I was like, you little stinker. Oh, you got me. And I was like, okay, you're just doing what you're doing. I didn't
and they
squish them or
can
anything
see
like that,
a lot more colors than we can.
And they have 7 color receptors, something like that. More colors than we do. And they can swivel their eyes independently.
What did you do to that mantis shrimp?
Nothing. I just watched them for a little while. And my anger cooled off, and my pain subsided a little bit. But
I wonder what Dr. Robert Carter tasted like. We could ask him.
No, he didn't get a piece of me. He just stabbed me. That's all.
Okay.
So, back to the color story. Red. I had bought a red soft coral. It's about as big as a silver dollar, and it's this squishy red thing. It's like a mushroom. In fact, it's called a mushroom coral. And it doesn't have a skeleton, but it's really leathery. And I bought it at an aquarium store in Miami. And usually I'd walk into an aquarium store and I said, hey, this specimen doesn't look so good. Can I buy that for you for $1? And they're like, sure. And I got and I bring them back to my aquaculture facility and nurse them back to health. Anyway, nice. Sorry guys, you got scammed. Anyway, I had this red thing, and I took a razor blade and I cut out a piece of pie, and I chopped it up, and I extracted the RNA, and I put it into E. coli. And this took a little longer to develop than the green did, but 48 hours later, I had one red spot on a plate, and it was like lipstick red.
Oh.
And my mouth is hanging open. I'm holding this plate in my hand saying, this is $1 million.
Oh.
No joke. It would have been $1 million. But I was holding a paper in my other hand that had come out that day. Red fluorescent protein.
No way.
Some guy in Moscow, like Russia in the 90s, impoverished Moscow.
Yeah.
Went down to his aquarium store and bought a little red soft coral that probably came from the same Indonesian village that mine had come from 2 years prior. And he'd been working on that for all that time. And the genetic sequence, when I sequence my gene, the genetic sequence is exactly
you discovered it a lot
same.
faster than
a whole lot faster. And oh well. At least I was right. I was right.
That's right.
So I graduated.
Yeah, see, this paper confirms I was right.
Well, it was the coolest thing, and to work in a field that was exploding. I mean, all these papers being published on fluorescent proteins and how important they became. I wouldn't have been able to do all that work. And I would have been very frustrated with my PhD because all the people, what does this do? What does that do? And I wouldn't have known because I'm just one guy. But all these other papers are being published. I'm publishing papers in the field too. And I was in a very comfortable position considering that I don't believe in evolution. And I didn't while I was in graduate school. Actually, I went to graduate school uncertain, but they unconvinced me about evolution within about 6 months. Interestingly. Anyway, so I'm there in a very awkward position, and I found out myself in the perfect field. something that grew really quickly, something I was really good at, and something that was really interesting and really exciting, and no one knew about it. Because a little secret for people out there who might want to get into science, yeah, go for it. Get yourself your bachelor's, get yourself a master's, get yourself a PhD. But the secret of getting a PhD is when you get to the point where no one on earth can tell you that you're wrong, that's when they give you your degree. And so what happens is you learn all the stuff about marine biology, and then coral reef ecology, and then Caribbean corals, and then coral proteins and coral genetics, and the green fluorescent protein from these 2-3 species that I studied. And I knew more than anyone else in the world about almost nothing. And so you drill down, you focus to this very specific little thing.
Wow.
And when you become the expert, that's when they say, congratulations, you passed the test, and they hand you the piece of paper.
Well, that's how you advance science. That's how you push the world forward.
One little discovery at a time. But I was bored stiff. Even though it was exciting and I got to scuba dive a lot, and I mean, it was a good life. But doing DNA extractions again and again, and growing E. coli and using the autoclave, and it was mind-numbing, and my brain was like, I can't do this anymore. I'm going to go insane. And so finally I got done.
You know, all this time I was just listening to your story, and I was realizing there's not much that we had in common with our education. I was home educated, and then I pursued a couple of different apprenticeships. I did some things outside of high school. It was fun. But one of the things that arose was going into filmmaking. I knew at that point that I would never go into filmmaking if I had to do it the old school, if I had to use real film and cut film and get into the chemicals and measure tape and splice these things together and develop film and sync it to sounds and nurse and baby film. I would never have gone into film. The turning point was I was about 14 and Apple was saying, oh, we can make movies with our computers now. And it negated all of the old school technologies, which were like laboratory experiments and systems. And
yeah. So what was the first movie that came out? It was all digital.
I don't remember. Well, there were several films that went back into the mid 90s, but they were mostly film festival films. I know that George Lucas was responsible for pioneering these for bigger budget films.
Yes.
Digital cameras technology. He's the guy who created not just Star Wars, but also the companies that made Hollywood special effects and technologies.
Yeah.
So they made a company just for sound. And then that sound company spun off multiple products and other sound engineering companies. But I think it was probably, I know they were talking about it when they were doing episode 1, but I know episode 2, Star Wars, was
Attack of the Clones.
Mm-hmm.
Ah, so that was the first big budget Hollywood movie that was just all digital?
Yeah.
That explains a lot as far as what we're seeing on screen.
Yeah. It has certainly changed a lot because of that. And there were other films that were big too, but not as big as Star Wars. I mean, you can't get bigger than that.
No.
But that was in like 2002.
Wow.
Digital was what convinced me I could go into filmmaking. And I was like, okay, I can do this. I will. I just will, because I know it's going to be so much easier than fiddling with all the chemicals.
Well, I'm glad you did.
Mm-hmm.
Because
Me too.
You've done some good work.
I couldn't have gone into audio engineering either. I couldn't be doing a podcast if we were still doing it the way that a lot of scientists do it in the lab.
Wow. Yeah, well, like my work in genetics now, it's all digital. I don't extract DNA. That's all done by machines anyway. I don't. What I do is as long as I have an internet connection, I have access to databases of DNA sequences, and I can download them and run computer programs and do analyses.
You know, I've never thought about that all the way through, but now that you mention that, I remember hearing you talk about various things that you are currently working on, and I'm like, well, how are you doing these experiments? There's not like a lab. You don't have a lab at home. You don't have a lab at the office.
Nope.
Wow.
Just a couple of weeks ago, there was a paper that came out. They just did 5,000 full human genomes from Singapore.
You said 5 or 5,000?
5,000 brand new, high quality.
Whoa.
Just from Singapore.
Wow.
So we are swimming in data. There's so much data, no one knows what to do. And it's a really cool place to be in the world of genetics now. And it's cool to be someone who's a little bit contrarian to the standard way of thinking, because there's some low-hanging fruit out there that we can pluck.
Mm-hmm.
Because not everyone's thinking the way I'm thinking. And we can look at it and say, wait a second, that's not supposed to be true. And boom, we take it into our fold and say, this is our territory, guys. Either disprove us or take a hike. And it's really cool. And we've actually done some pretty significant advances in science just because we think different. And I say we because I'm not alone. I'm working with several other people in our little creation genetics group.
Interesting.
Yeah.
So what we got here is your career in the past as a marine biologist and how you got there. But we're right on the cusp of when you went into genetics. And I think that's where we got to stop for today.
Oh, okay.
Yeah, so this is going to be another two-parter. We're going to have to talk about genetics, biblical genetics at that.
All right. So Adam and Eve, Noah's flood, Tower of Babel.
But also how you
people.
wound up in genetics and what your research has been up to the present.
Okay.
It's really interesting stuff. I love this stuff. And it's interesting, too, how marine biology and genetics, I mean, I wouldn't have thought that how they could be paired together, but it's your career path. It's fascinating stuff.
My career path was laid out for me before I found it. So someone out there who's like, I want to go into education. I want to be a scientist, or I want to get a high degree. What do I do? You know what I tell people? Go for it. See how far you get. You don't know what doors are going to open, but they're not going to open unless you go knocking.
Mm-hmm.
Just go. Now, I don't want people to have, $10,000 or $100,000 worth of student loans and no possible way to pay it back. So I tell people, when I say, I'm going to be a biologist, I say, why? You want to flip burgers the rest of your life? So you're at least going to get a master's degree. Don't waste your time by getting a degree in biology. It'll do nothing for you.
Oh, interesting.
Like, art history. Why are you going to do that? Yeah, it's interesting, but how many art historians are there in the world? What's the job market? It's bad. You're going to be doing something different. So, okay, get a degree, fine, go do something different, fine. But if you want to be a biologist, get a master's, get a PhD, if you can. But see, as a Christian, I had to resolve that I wasn't going to lie if someone asked me what I thought about, evolution or something like that. Not that you have to be, anti-evolution to be a Christian. But from my position, I said, I'm not going to lie, but I'm not going to volunteer. I had a goal. I wanted those 3 letters after my name, and I didn't want someone to unfairly target me or torpedo my chances. And so I never lied. Nobody ever asked. Nobody cared. And as long as I was a good student, and as long as I was, seemed like I was towing the party line, I wasn't lying to anybody. And I, carefully crafted my answers in my essay questions in classes. And my doctorate was difficult because they were demanding I do some evolutionary things in my doctorate. So all I did was I said, okay, you want evolutionary stuff? Fine. And I said, here's all this evolutionary stuff, and here's all the contradictions to all this evolutionary stuff. What do I know? I'm just a graduate student. And I didn't say it that way, but I got compliments for those sections because it was a penetrating analysis, and I wasn't just, taking the evolution is true, therefore evolution is true. No, I was saying, yeah, but check out this, and how do I answer this? And this is something to wrestle. And it was worth really digging in, and it was a lot of fun.
That's awesome.
So maybe that'll be another episode. How do you survive graduate school or college if you don't believe what other people believe?
Yeah.
That's a cool question.
An idea related to that would be just a journey for the student to scientist.
Yeah.
And you could give great advice.
Yeah.
So thank you so much for joining us on this quest. If you want to dig deeper into this episode's topics, you can find links to the articles and the like of anything that Rob has done in his career or he could refer to that would be of interest to you and the bioluminescent fish and coral. Interesting stuff. Do you have a picture of such examples online that the audience can see some of your fish or corals or anything luminescent? That'd be great.
Oh, yeah. Okay, we'll put them in the show notes.
So if you want to see those things, you can find them in the episode show notes at our website. That is nightowl.fm/equinox/11 for this episode. And if it's more handy for you, you can also find the show notes, which are available with this episode in your podcast app. If you want us to discuss a science topic that you have in mind, please tell Rob at Biblical Genetics on Facebook or YouTube, or you can send a tweet to @podcastequinox and we'll get your idea in the queue. If you haven't already, go to Dr. Robert Carter on YouTube and Facebook. Just look up Biblical Genetics and watch his latest and join the discussions. And you'll find me on Twitter if you want to catch me. I'm at JCS Darnell. Until next time, goodbye, Rob.
Goodbye, Joe.
And thank you, dear listener, for listening to Equinox.